A mutation is an alteration in the nucleotide sequence of a genome. This can occur in that of an organism or virus, and they vary in both length and severity. Just as if you are copying notes in school and you make a mistake, your mistakes vary. Sometimes even with these mistakes, you are still able to understand the message or information copied. Other times, you cannot. This is what happens in a cell at a submicroscopic level. As seen on this chart, some of the different combinations of nucleotides result in the production of the same amino acid and therefore the same polypeptide sequence. This is a silent mutation, so even if the sequence has been transcribed wrong, the result is still the same. But this is not the case for all transcription errors. Some cause drastic changes within the cell, known as missense mutations. When a mistake results in the generation of a codon that produces a different amino acid, it can lead to a non-conservative mutation where the product differs from the original sequence. Now, why are we even talking about mutations in the middle of a pandemic? As I said before, mutations can occur in the genome of a virus. COVID-19 is an RNA virus. It is spread through respiratory droplets that travel from one host to another when a person coughs, sneezes, or talks. Once the virus has hijacked the host cell by penetrating the cellular membrane, the viral capsid is dissolved and the genetic material is released inside the cell where it begins the process of replication. Using the host cell's RTC, the virus uses the enzyme RNA polymerase to link nucleotides together to form a new RNA strand from the original strand. This is where problems may arise. The difference between DNA and RNA polymerase is that DNA polymerase proofreads the transcribed sequence and RNA polymerase does not, leading to far more errors in transcribed RNA. As viruses use the machinery of the host cell, the pieces of uncoded virus float around the cell along with pieces of the uncoded genome that were originally in the host cell. While investigations are still ongoing concerning COVID-19, looking back at past outbreaks, they began with the nucleotide sequences from an animal virus and a human virus, combining in the cell of another host animal that was able to contract both viruses. With parts of both viruses floating around, a non-conservative mutation occurs, enabling a new virus to infect humans. Mutations are part of the viral life cycle and are bound to happen. Big or small, viruses change with every generation, causing what is called a strain. If a change is successful in infecting more people, it will spread quickly, and therefore that strain of the virus will become more common as cases multiply. This follows Darwin's theory of evolution, where he explains natural selection, where organisms with heritable traits that favor survival will leave more offspring than others, causing their traits to increase in frequency. The same goes for the reproduction of viruses. One challenge of mutations is that it can make the creation of a vaccine more difficult. This is why there's a new flu shot every year. Researchers look for patterns and ways to get ahead of the virus so that it can be oppressed by the time it arrives. However, these mutations can actually also help us in our research of the novel coronavirus. Since mutations change the virus's genetic material that is passed along as it spreads, mutations allow researchers the ability to track how the virus has spread. By the samples taken from patients across the world, researchers create a family tree to show the relationships among the strands found, which allows us a better understanding of how this virus spreads and what we can do to better protect ourselves and those around us. So, while there are still so many unanswered questions, we can use the information we already have to make the best choices and to better understand the world we are currently living in. Thank you.